Today we're going to be talking about RNA mostly. So just a very quick review of DNA and how DNA is synthesized or made, and then we'll move on to the types of RNA, and that'll set the stage for how it contributes to protein synthesis. So remember, DNA we now know is this double helix shape, right? Kind of like a spiral staircase, right? It has some sugar and some phosphate, sort of in these pinkish purple areas. And remember, pretty straightforward. A and T's always go together, adenine and thymine, and G and C's always go together. I always remember at Cottage Grove, right? So A and T at Cottage Grove, C and G. So those would be these rungs, the rungs of the ladder. Okay, so just sort of another view of it there, a little more space-filling view. So we know we have a sugar, right? Ribose sugar. Here's your phosphates, sort of like when we talked about ATP, right? So remember our macromolecules, and then these colors would be our nitrogen bases. So DNA replication here, if you look at it, shouldn't surprise anybody. Lots of enzymes involved, right? So we have one called a helicase, which is the one that cuts the helix, and we have another one called a polymerase, another enzyme, the polymer, makes a polymer, is helping rebuild the DNA. Remember, this is a semi-conservative process, semi meaning half in this case. And so if you see the old DNA, here's an old fork of the old one piece of that old DNA, here's another old fork. And so each of those old forks gets a new section added onto it. So it's literally half and half old and half new DNA. And at the end of it, you've got two copies. And you can see that back here, the chromosome. And obviously we'd be looking at this in the nucleus. Okay, I will be asking you to do this on a formative quiz, right? Match these up. And again, it's pretty straightforward with DNA. There's a little addition that gets added with RNA, but at this point, remember CG, AT, CG, right? GC, TA, right? So just these bases like this. And remember, in a human, the human genome has roughly 3 billion of these, right, in every one of your cells. So lots and lots and lots of them in every cell. That's why it's 10 feet long in every one of your nuclei. Okay, so in order for DNA to make a protein, it first has to be decoded. Okay, so RNA is really the carrier or the decoder. It's another type of nucleic acid, so it's another macromolecule, but it has a few differences. Number one, the sugar is slightly different, right? So instead of a deoxyribose, we have a ribose sugar. No big deal. But here's one significant difference. Here in DNA, you can see you have adenine, cytosine, guanine, and thymine. But in RNA, the thymine gets replaced with something called uracil. Otherwise, the other ones are all the same. Another significant difference is that DNA is, of course, double-stranded, leading to the double helix, but RNA is only single-stranded. Now, there's several types of RNA, and that's what I'm going to touch on here next. So, the main function of any RNA is to play a role in protein synthesis, so the helping of making proteins. It controls the assembly of amino acids. If you remember, those are the monomers for proteins. And those, of course, form proteins. So there's three major types. One is called mRNA, and the M stands for messenger, and it's useful because that's exactly what it is. It acts as a messenger between the DNA in the nucleus, right, which can't leave the nucleus, to the cytoplasm. So it carries information as a messenger from the nucleus out into the cytoplasm. Once it gets out into the cytoplasm, it interacts with a ribosome, and ribosomes are actually made of RNA, so it's called rRNA, ribosomal RNA. And this is where I hope most of us know proteins are assembled. So these are the little assemblers out in the cytoplasm. Remember when we talked about the rough endoplasmic reticulum? The endoplasmic reticulum is rough because it has ribosomes studded all over it. Okay, so right outside of the nucleus, the mRNA carries this information out to the rRNA, which is embedded in the endoplasmic reticulum. And then another one, the final one, is tRNA, and this is called transfer RNA. And it's actually going out into the cytoplasm and finding the needed amino acids to match up with what the mRNA is telling it so that the proteins can be built in order, right? So these amino acids can be strung together to make proteins. So tomorrow we're going to look a little more in detail at the transcription process. And if you remember, trans means to go across. Script, I think we would all guess, has something to do with writing. And so writing across, we're going to look at that in a little more detail. And then the next day we get together, we'll be talking about translation. So those are the two big parts of protein synthesis, and they have a lot to do with these types of RNA. All right, see you next time. And again, remember 2 o'clock today if you'd like to join us for a meeting. If you have questions, that type of thing, from 2 to 3, just click on that WebEx link and you can join right in.